All right, welcome to another deep dive. Today we're going really small.
Yeah.
Like really, really small.
Microscopic.
We're talking like cilia, right? Those hair-like structures you find all over cell surfaces.
Exactly. Silly.
But we're going even smaller than that.
Even smaller.
We're zeroing in on the protective layer around cilia.
It's called the Glycocalyx.
The Glycocalyx, yeah. We're trying to figure out how this this outer layer, I guess you could call it.
It is. It's the outermost layer of the cell.
Influences how cells interact,
Like their environment, yeah.
with their environment. And our main source material for this is a brand new paper, hot off the presses, from Advanced Science, just came out this month.
Yeah, March 5th. It's a really interesting one.
So what makes this research so interesting?
Well, there's a team, right? This German and Italian team. They created this really, really detailed map.
Mm-hmm.
And it's of the Glycocalyx in a green alga. It's called Polendomonas reinhardti. It's kind of a mouthful, but basically this map, right? It's like a blueprint, giving us this unprecedented insight into how these super tiny structures, how they affect how cells stick together, how they move, even how they sense what's around them.
Okay, so Glycocalyx.
Yeah.
I'm just trying to like picture it in my mind, Is it like a sugary shell?
It's pretty close, actually, yeah. The Glycocalyx, at its core, it's made up of these things called glycoproteins, so proteins, but they're decorated with sugar molecules.
Oh, okay.
And this layer, this Glycocalyx, it acts like the cell's first point of contact with everything outside of it.
Oh, interesting.
So, it's vitally important for any kind of interaction the cell's going to have. And, up until now, the actual precise structure of it has been kind of a black box.
Oh.
So this is really breaking new ground.
So basically what they've done with this alga is they've given us the blueprint for what this Glycocalyx actually looks like.
Exactly. And from this blueprint, they identified two major glycoproteins. One we already knew about, called FMG1B. But then there's this other one, a variant they discovered, FMG1A.
FMG1A.
Yeah, and here's the thing. These glycoproteins, they're similar biochemically to mucin proteins.
Okay, now mucin proteins, they sound familiar. Don't those like make mucus slippery?
Yeah.
Like in our noses or lining our organs.
Exactly. You're spot on.
Oh, okay.
So mucins, they're glycoproteins too, right? They make that slippery, that protective barrier you see in mammals and other animals. And the fact that we're finding these similar building blocks in this alga on the cilia, it hints that maybe there's some evolutionary reason for why these protective layers are formed this way.
Oh, that's fascinating.
Yeah.
So, they figured out the blueprint.
Yeah.
They found these proteins.
Yes.
And they're similar to what we find in mammals. But then here's where I think things get really interesting, because they didn't just stop there. They did this experiment where they actually removed these FMG proteins, both of them.
Right. They wanted to know what's their function.
Right. And what happened?
The results were kind of a surprise. The cilia became a lot stickier.
Really?
Significantly stickier.
Wow. But I thought those proteins were what made it slippery.
Right, but here's the thing.
Yeah.
Even though the cilia were stickier, the algae could still move.
Wait, so if removing these proteins made the cilia stickier, but they could still move, doesn't that mean those proteins aren't actually the main thing making them stick or giving them the power to move?
That is exactly the conclusion that they came to.
Yeah.
Yeah. So, we thought these glycoproteins were like the glue, right?
Yeah.
Actually, they're more like a non-stick coating.
Oh, that's a good analogy.
Almost like, Teflon or something. And this coating, it can be adjusted to make the cilia more or less sticky as needed.
That's wild. Like it completely flips our understanding of how those things work.
Yeah. It's a real paradigm shift.
And, Professor Michael Hippler, one of the researchers on this, he said that this completely changes the way we think about how cells manage their interactions with their surroundings.
Right. It's not a passive thing. It's an active process.
And, this has got to have huge implications for, all sorts of things, right? Like how cells communicate with each other, even maybe how infections happen.
Absolutely. And, Dr. Adrian Nievergeld, another one of the researchers, he pointed out this could give us clues about similar protective strategies in other organisms, not just algae.
Oh, I see. So it's not just about, this one type of alga. This research could apply way beyond that.
Right. And I think another really cool thing about this study was how much collaboration went into it. You had the University of Münster, the Max Planck Institute, and Dr. Gaia Pagino's group over in Milan, all working together.
It's an international effort.
Yeah, it was.
And they used, I mean, some really amazing technology to figure all this out, right?
Yeah, absolutely.
Cryo-electron tomography, and, I mean, I've heard of mass spectrometry, but that sounds incredibly complicated.
There are very advanced techniques. Cryo-electron tomography.
Yeah.
You know, imagine being able to take a 3D snapshot of these tiny structures, super, super high resolution.
Oh, wow.
And then the mass spectrometry, it lets them identify the proteins with like pinpoint accuracy.
Yeah.
And of course, they use genetic techniques to actually remove the FMG proteins in the first place.
Right.
You know, without that whole combination of tools, they wouldn't have been able to paint such a detailed picture.
Yeah. Okay, so just to kind of sum up here.
Yeah.
The Glycocalyx in this alga, it's made of these two proteins, FMG1B and FMG1A, which is the new one they found.
Yeah.
But it doesn't directly cause the cilia to stick or move. It's like a regulator. It controls how sticky they are, and it provides a protective layer.
Exactly. It's a whole new way of thinking about how cells interact with their environment.
So that begs the question,
Yeah.
how does this concept of a regulatory protective layer, how might that connect to other biological barriers, other interfaces that, you know, we see in nature?
Yeah. Think about your lungs, right? The lining of your lungs. Or the surface of a plant leaf. They have to interact with their environments too.
That's true.
How are those interactions managed? What kind of mechanisms are at play there?
That's pretty mind-blowing to think about. It
Yeah.
really makes you appreciate just how complex life is, even at the smallest scale.
Definitely. There's always more to uncover.
Well, thanks for taking us on this deep dive into the micro world.
My pleasure. It's fascinating stuff.
It really is.